Okay, great. Hello everyone. It's been a long time since the last video made by me. We've covered a lot of material since the last midterm. Retroviruses, HIV, herpes, vaccines, and we'll have a bit more this last during that week. So for today, we're going to do retroviruses. And honestly, this section was the most difficult for me. There were a lot of mechanisms and so forth. So I plan on drawing a lot of pictures in this. So I think this video will be cumulative for retroviruses. It will be a bit over an hour, but I think it will be a good hour if you decide that you want to watch. A lot more pictures so you don't get a huge wall of text. I'm hoping that the pictures I draw will be helpful. Okay, yeah. Alright, so let's get straight to it. Like always, any virus, first thing you want to do is lower characteristics. Okay, so first thing you know is that this is a positive single-stranded RNA virus. Just like polio, I guess, in the sense that it has a positive single-stranded RNA genome. What makes it special and different from picornaviruses and pretty much any other viruses is that it uses a reverse transcriptase to convert from the ssRNA to a normal double-stranded DNA and then back into positive single-stranded RNA. That's why there's the word retro, as in you do the reverse. You do a reverse transcription. So normally transcription you do DNA to RNA. Now you do the reverse or reverse transcription in virus. Okay. Anyway, so it's positive single-stranded RNA. Its envelope. And its envelope has glycoproteins that help in its attachment and penetration into the host, into our human cells. This thing is 100 nanometers, so it's pretty freaking big. Okay, so it's just straight copy. And you have a helical nucleocapsid. Wait. A helical nucleocapsid, yes, nucleocapsid. But what's kind of confusing is in lecture it's helical, but in the book, the picture, and even the nucleocapsid is, you know, the material that wraps around the DNA, but it has a icosahedral shape to it. I'll draw a quick picture just to illustrate it, so just bear with me. Icosahedral capsid. So helical nucleocapsid, but also has an icosahedral capsid. So not nucleocapsid, capsid. And it has the VAPs. Remember, if you recall, VAPs are the virion-associated polymerase. And the polymerase is reverse transcriptase. Which is abbreviated RT. Okay. Next thing to know about it. It infects lots of stuff. Affects many groups. So these groups include stuff like chicken, rodents, and humans. Or chickens, rodents, cats, or scientifically felines, but in terms of common, I guess, ordinary speech, it's cats and humans. So essentially, you can eat chicken, but you should cook it please don't eat mice because they have a lot of diseases. Cats are our pets, don't eat it, but you know, you also, if you do eat it, it has retroviruses, so maybe you should cook it. I hope none of you are thinking about eating humans, just, even if there weren't any retroviruses in us, it's immoral, okay? But if you do decide to eat human, just cook it. Alright, anyways, moving on. And some important discoveries. Okay, the reverse transcriptase, which I'm abbreviating RT here, was discovered in 1975. Proto-oncogenes. Which are also called C-oncs or V-oncs. It was discovered in 1989, and HIV was found to be a retrovirus in 2008. So with this book right here, Proto-oncogenes, it's the C-oncs stand for cellular oncogenes, V-oncs are viral oncogenes. And what's important here is that in our body, we have genes called oncogenes, and they basically tell our cell when to grow. Excuse me. The only thing is that it's regulated. You know, you want your cell to grow at certain times, you know, like during childhood and puberty, I guess. You know, you're growing bigger. So you'd want your oncogenes to be turned on. But the V-oncs are viral homologs to our cellular oncogenes. So they do the same thing, but they might be structurally very ever so slightly different. But essentially what the V-oncs do is they tell our cells to grow uncontrollably. And by growing, I mean, you know, synthesizing protein, making DNA, dividing, and so forth. Another thing about V-oncs, though, is that sometimes they aren't actually really a cellular homolog of sometimes they aren't a viral homolog of a cellular oncogene, or rather there's something, it would be a gene that messes up the cellular control mechanisms of a cellular oncogene. So tying this back into retroviruses, what makes retroviruses, one of the key characteristics of retroviruses is that they can cause tumors. And they cause tumors because they have oncogenes. And sometimes it causes tumors not by like an actual gene, you know, like polyomiviridae or papillomiviridae. You know, those have like actual genes that did stuff. Sometimes the proto-oncogenes here, the oncogenes, sometimes the retroviruses cause cancer simply by inserting its DNA next to a normal functioning cellular oncogene. But because there's the extra new sequence of foreign viral DNA next to that oncogene, now you get a tumor. So I think as we go through retroviruses a bit more, you'll see what I mean. Also, third bullet point, HIV is found to be retrovirus. Even though Dr. Samuels covered HIV separately in class, and the book covers it as a separate on subject matter, it's technically part of it. Actually, not even technically, it is part of retroviruses. But the only reason why it gets special treatment is because it causes a lot of human suffering. And it's a very important pathogen in terms of human pathology. So stop this if you need to, I'm going to erase it.